Hi, I'm Vaidyanathan, and I'm here representing my research group at Sheffield in the UK. We're currently focused on developing an understanding of carbon dioxide fixation by microalgae and the resulting value additions. I'm here to discuss the concept of developing microalgae-based carbon dioxide fixation to drive the bioeconomy. So this would be classified as new concepts and emerging technologies within the context of the symposium. Biological carbon dioxide fixation can be achieved with a better understanding and utilization of the natural phenomenon of photosynthesis, where carbon dioxide is fixed to reduce runoff of carbon in biomass and products with the help of reducing equivalents from water and light energy. The reduced runoff of carbon in biomass and product could result in products of economic value, and these include as energy vectors. The key is in the use of sustainable resources to enable the process. Every mole of carbon dioxide can store about 114 kilocalories of free energy within plant biomass, and every kilogram of algal biomass can result in the sequestration of 1.9 kilograms of carbon dioxide based on the stoichiometry that is shown here. Biomass productivities of 30 tons equating to carbon dioxide capture of 50 tons per hectare per year are feasible with current technologies as conservative estimates. Higher numbers can be seen in literature, quoted in the literature. If you look at microalgae as photosynthetic organisms, they're quite diverse genetically as well as by habitat, showing high metabolic versatility. Some members of the group of cyanobacteria and green algae can produce hydrogen, albeit at levels which are still not practically significant. Oil from accumulated in algae can be used to produce biodiesel, but this still requires sustainable processes, economically sustainable and economically competitive processes to be developed. Algal biomass can be grown as feedstock to accumulate fermentable sugars to produce alcohols through fermentation. Similarly, biogas production by anaerobic digestion of biomass feedstock is possible, as well as direct use of biomass for bio-oil production by thermochemical routes is also possible. The question to be asked is, how efficient are these processes? And how much of carbon are we able to fix? Can this be maximized? There's currently several products in circulation in diverse sectors where carbon dioxide has been stored through microalgal cultivations and routed to products of value. However, the question we need to ask is, can this be developed towards sustainable circular bioeconomy? Fixing the carbon in itself is not sufficient. It is important to generate value additions and value productions which are needed to enable sustainable propositions. There is a drive to meet the economic viability of any proposition to produce algal biofuels by doing so in a biorefinery to incorporate additional valorizations. Biorefineries enable multiple products to be obtained from the same chassis or process. For example, 6 products that are shown here for a diatom biorefinery. The objective is to maximize the number of products or value additions per chassis for every single chassis or run. The idea is to maximize carbon dioxide fixation in biomass, which can be routed to products of value within a biorefinery, and these can include energy vectors that are either non-carbon based, for example, hydrogen or carbon-based energy vectors, which on combustion would enable energy generation and release of carbon dioxide back into the atmosphere. A slower release of trapped carbon dioxide in bioproducts after sufficient value generation is desirable and is ideally desired. It is required to develop circularization of not only the carbon, but also the resources used to fix the carbon in order to generate an economy that is sustainable. There is a need to develop a better understanding of carbon uptake or carbon dioxide fixing capacity by these organisms, which we are focusing on, and we have been looking at storage products, in particular lipids and carbohydrates, and ways to maximize oil accumulation. We are interested in understanding and maximizing carbon dioxide conversion to a fixed carbon of value. And we're also interested in knowing, can we increase the throughput of carbon from carbon dioxide to products of value, as well as can we uncouple growth and accumulation of products for sustainable productivities, because often growth and product accumulations are inversely proportional. We're also interested in other aspects of microalgal cultivations. Carbon dioxide supply does not necessarily equate to carbon dioxide availability and utilization. So microalgal cultivations predominantly are grown as suspended cultivations. So there is a requirement for carbon, which is in the gas form, to be dissolved into the liquid form, in aqueous form. And further equilibria define the type of species that would be present. The Jerome plot shows a major role that is played by pH, temperature, and salinity to determine the type of species that are available for fixation. So gas has to be solvated, solubilized on supply, and equilibrated in liquid between different species before it is available for uptake by the algae. Kinetics of carbon uptake is an interplay, therefore, of chemical and biological equilibrium, and the process is dynamic, requiring further understanding and increase efficiency of uptake. Our estimates indicate that current efficiencies of carbon dioxide fixation in air is less than 20%, and this is dramatically reduced to less than 5% with increase in carbon dioxide of increasing concentrations of carbon dioxide in the inlet gas, leaving considerable scope for research and development. Carbon assimilations do not necessarily translate from availability, as can be seen in the figure. Increasing the supply of carbon dioxide in the inlet gas from air, which is of the order of 0.04% to 6%, does not necessarily, at the same gas flow rates, only results in a marginal increase in biomass formation and concomitant fixation of carbon dioxide. Further increase to 20% carbon dioxide drastically flattens growth due to decrease in pH. Speciation of carbon to different species at different conditions can influence uptake, and pH has a significant role to define the species that is present at a given condition. Oxygen in air is inhibitory. So when we replace oxygen in air with nitrogen, for example, for the same amount of carbon dioxide in nitrogen as opposed to carbon dioxide in air, we can see threefold increase in growth rates, as well as threefold increase in multiple fold increases in carbon fixation rates. Carbon supply dynamics can enhance carbon fixation rate, and this can be used to develop strategies to maximize uptake. 100% carbon dioxide is often inhibitory to carbon uptake by algae. However, this is not algacidal. So the process is reversible. So on supply of air, the organism is able to take up air. Therefore, this could be used as a strategy, intermittent passage of higher amount of carbon dioxide to maximize uptake rates is possible. A key area of development is the sustainable sourcing of carbon dioxide. Carbon dioxide from microalgal cultivations can be sourced from either the atmosphere or as well as from, more importantly, industrial emitters of carbon dioxide. So we could provide a solution for the current problem with respect to industrial emissions of carbon dioxide. So direct capture, air capture in its simplest form, can be employed directly to algal cultivations, or the carbon dioxide in air could be concentrated to higher levels and then supplied for microalgal capture. The challenge here is to make the carbon dioxide available in an economic and environmentally sustainable manner. So carbon capture from direct air capture can result in carbon dioxide concentrations of 0.04%, which is present in air, to 98% present in concentrated streams. Equally, we can utilize carbon dioxide from strong carbon emitting industries such as power plants, ammonia production, and cement manufacture, where the carbon concentrations range between 6 to 60% and beyond. These are non-biogenic carbon dioxide that is available, but equally biogenic carbon dioxide is also available. For example, from ethanol production, where every mole of ethanol that is produced results in a mole of carbon dioxide that is formed, and this carbon dioxide is 100% pure. So 100% pure carbon dioxide can be used to be captured by microalgae and then for further value additions, conversions to other forms of carbon for value additions. Equally, anaerobic digestion can also be used where the carbon dioxide occurs in biogas. So biogas is a mixture of methane, carbon dioxide, and hydrogen sulfide. So algal cultivations can be used to cleanse biogas of carbon dioxide, and that carbon can be routed through algal cultivations to products of value, to biomass that could then be used for anaerobic digestion itself in a circularized process. Similarly for ethanol production as well. In all these cases, a capture step might be required. So the carbon dioxide concentrations are of the order of 30 to 40% in anaerobic digestions in biogas. So the carbon capture step, an intermittent carbon capture step might be required to buffer the gas compositions or gas concentrations to enable effective capture by microalgal cultivations. The fixed carbon should result in marketable products to enable a sustainable bioeconomy and circularization of not only the carbon, but also of the resources within the to enable a sustainable economy. And that is the proposition we put forward. Thank you for your time, and thank you for listening.